Okay, so you already learned about DNA, and DNA is made into genes, and the whole point of genes is to make proteins. Proteins do pretty much everything in our body. They make up all our structures. They do all the chemical reactions through enzymes, their hormones, just on and on and on. So proteins pretty much do everything in our body, and they're all coded by a different gene. So that's why DNA is important. That's why proteins are so important. The only thing that proteins don't do is give us energy. Remember, carbohydrates give us energy. But so what we have to do is we have to build our own proteins in our body. And we do that by something called protein synthesis to build proteins, and we use genes to do this. So what we're going to do is learn how we get from a gene to a protein. All right, so we're getting from a gene to a protein. So if you remember from the last segment, and I'll just write this down here, we had that organizer. We had DNA goes to a gene. And remember, a segment of DNA is just a gene, it's just a sequence of bases. So that's what we talked last time. Then we had this, where DNA goes to a protein, and that's what we're going to focus on here. And then a protein determines our traits, and that's what we're going to more focus on in the next section, the next unit. So right now we're going from here to here. How do we get a gene into a protein? So it's in two steps, transcription and translation. So again, the whole process of protein synthesis, its goal is to put a bunch of amino acids, because remember, amino acids make a protein. And we do that through two steps, transcription and translation. Today we're going to just focus on transcription and also give you a little bit of background. So let's look at our notes here. So transcription is the first step of protein synthesis. Protein synthesis is putting together amino acids to form a protein using a gene for the code. And the two steps are transcription and translation. So we're going to focus all on transcription and do some activities on that first, and then we're going to go to translation. But before we go into those, we need to give some key background information. So we have three key vocab terms that you're going to have to know, and then we're going to talk more about this RNA stuff. So we have two key nucleic acids, DNA and RNA. And the thing you have to remember about DNA is it's found in our nucleus, and it has to stay there. It can't leave the nucleus. But proteins are made out of ribosome, which are outside the nucleus. So we have to find a way to get the code of the DNA from the nucleus to the protein, to the ribosome, and that's where RNA comes in. So DNA is just the sequence of bases used to code for a gene, and has to stay in the nucleus. It's too big to leave. All right, the gene is a sequence of bases of DNA. I'll say of DNA, I'll change it a little bit, that codes for a protein. All right, so DNA makes up our gene, gene codes for a protein. And then RNA is the structure that it's like the transition structure. So kind of like going from one piece to the other, this is like the middleman. So this would be the Walmart, basically. You want food, the farmers make food, Walmart's in between, and they sell the food. So RNA is our mediator, our middleman. So RNA is the transition between the gene and the protein. So what we have here is we have our nucleus. So what I want you to do is label in here. So in our nucleus is going to be, this is going to be our DNA and gene is going to be in here. And then what's going to happen is our RNA is going to move out to here. So RNA moves, one type it does, and we're going to talk about the different types, but the RNA moves from the nucleus out here to a ribosome. Alright, so we're going to add a ribosome out here. Alright, so DNA genes found here, we're going to go to the ribosome, and then the ribosome is going to make proteins. And that's what's going to happen there. Alright, so what is RNA? Well, RNA is ribose nucleic acid. And there are three different parts of, or there's three differences between RNA and DNA. So first of all is the sugar. This is the least of the three that I want you to remember. So the sugar in the DNA is deoxyribose. That's where it gets its name, deoxyribose nucleic acid. And same with ribose. Ribose gets its name from ribose nucleic acid. So the sugar is different. And that's all. I don't really care too much about that. These next two are more important. So if you just look at these, what do you notice? Well, this is DNA. It's double-stranded. RNA is a single strand. All right, so DNA is double strand. And RNA is single strand. And then the last one, and actually we can just look at this picture down here. Our last one, if you look at these bases, they both have C, they both have G, and C pairs with G in both cases. They both have A, but RNA has this uracil, or U, instead of T. So A pairs with U in RNA. So U, RNA has U instead of T. So that's how you can tell. If you're getting a sequence of bases and you're trying to figure out is it DNA or RNA, you're going to look at that. The U will give it away. Alright, now there's three types of RNA. So that was just differences. Now there's three types. There's mRNA, tRNA, and rRNA. And the first letter tells you what it does. So we're going to start with mRNA. mRNA is called the messenger RNA, and its job is to take the message of DNA out here to the ribosome. All right, so that was the RNA that I was mainly talking about before. So this is messenger RNA, and it's involved in both transcription and translation, and it takes the DNA code from the nucleus to the ribosome. So what happens is, which we'll talk about in a second, is DNA is copied into mRNA, and then the mRNA copy goes out to the ribosome to make the protein. The other two, the rRNA isn't as important. RNA is just what the ribosome is made of. This only happens in translation because ribosome's only involved in that one, and it's just involved in building amino acids into proteins. Alright, tRNA is another important one. tRNA, so here's our ribosome, and it's reading the mRNA because that's the copy of the DNA sequence, so it reads it, and then it tells what amino acids come in, and what brings them in are these tRNAs. tRNAs are called transport RNA, or transfer, whichever one you would call it. I also call them taxi drivers because they just pick up the amino acid and drop them off where they're supposed to go. So tRNA, transport, move, transfer, and the T stands for like a taxi driver. Alright, these are only involved in translation, and they carry amino acids to the ribosome. Alright, they also kind of look like T's. If you look at these pictures, well, this picture isn't a very good one, and that picture not very good, never mind. But sometimes they look like T's. So here's the amino acid that it's carrying, and here's the mRNA, and so it brings it here, and then it leaves the amino acid behind to be forming as a protein. So this is the protein growing. Alright, so now we're going to talk about transcription. We actually skipped around a lot of the stuff that happens in translation. We'll talk that next. Transcription is really simple. Alright, really easy. What it is first of all, it happens in the nucleus because it involves DNA. And DNA can't leave the nucleus. Why does this happen? Well, we need to do this. We need to convert DNA into mRNA because DNA cannot leave the nucleus. Alright, it's too big. So we need to have a transition. So mRNA needs to carry the code out of the nucleus. Alright, what happens is DNA is copied into mRNA. Alright, pretty easy. So DNA, specifically the gene, is copied into mRNA. So let's look at this animation and we'll talk about the different steps. So step 1 is, in order for DNA to get copied, it has to unzip. Alright, so DNA gets unzipped. And then this RNA polymerase, which you don't need to know this, but this enzyme here is copying the DNA into mRNA. So step 1, DNA is unzipped. Alright, and you should draw this too, so I'm going to insert a quick picture up here. So insert image, and let's see if I can find DNA unzipped. Alright, so this is best. So this would be better to do on paper, to show the DNA breaking apart. Alright, so I'm going to attempt to draw this here. So step 1, we have our DNA code here. So we have our DNA. And then we would have our bases going across, our rungs, our bases. So remember, the outside DNA is your sugar phosphate. And then what happens when it gets unzipped is you end up with 2 straight lines here. And here's the bases coming off of one end. Oh, what's going on? Alright, so we have basically we broke the ladder in half. And we have the bases on one end based on this. So this might be A, T, C, G, G, C. Alright, so that's basically the first step. DNA's unzipped, and I know that looks like a little kid drew it. The second step then is just mRNA gets copied, and you can see G's pairs with C's, A's pair with U's. This was an A and G here. There's a C behind it. You can't see it. A pairs with U, C with G, C with G, C with G, A with T. Here, this one's a T on DNA, so it goes with A. But whenever there's an A in DNA, you show up as a U. So big, again, key, very important. Every time you see U, you know it's mRNA. So mRNA is one, it's single-stranded, and it's gonna have U's in it. So if I drew this again, so now just draw it like that picture, we have our DNA here, our double-strand DNA, and then out comes the mRNA. Actually, let's delete that. Our DNA comes out of here. Alright, and then remember, this is our mRNA. And this is our DNA up here. Alright, and then here's our, remember there would be bases on here, so I'm just going to draw like a little U to know that there will be at least some U's on this mRNA. And remember, single-stranded, U. Alright, and then finally, the last step, and you should draw these in more detail, is the mRNA comes off of it, and then the mRNA is going to go out of the nucleus. It doesn't show that on this diagram, but there's our complete mRNA. If you look at it, it's almost, this is that complementary to those bases. So that is transcription. So we took DNA, opened it up, made mRNA, and then mRNA leaves the nucleus. And I don't have time because of my video constraint here to draw that, but you should probably draw that. And that is it for this segment.